Okay, I am going to go over with you the steps of mitosis. So remember that mitosis is the division of the DNA, and then cytokinesis involves division of the cells. So a couple terminologies that I want to show you. So what I have here, I hope you can see these colors. I have two chromosomes that are yellow. All right, just to show you like pairs of chromosomes, so one from mom, one from dad. I have two that are red, one from mom, one from dad. And I have two that are blue, all right, one from mom, one from dad. So these just represent different chromosomes. In humans, we have 46 chromosomes, so we'd have 46 of these. So this individual, the diploid number here would be 6 instead of 46, because I want to simplify things a little bit. And these are chromosomes, and think of these beads as different sections of DNA that are genes that would code for a protein, like how to make a protein. All right, so each little section of this codes for, a gene that tells you how to make a protein. So, like I said, this would be the diploid number of 6. It doesn't really matter which ones are mom and dad's chromosomes for mitosis, but it will when we get to meiosis. So for right now, we're just going to say they are two different chromosomes. They look alike because they have the same genes on them. So like at this top part here, if this is the gene for example, eye color, there would also be a gene for eye color on this chromosome and a gene for eye color on this chromosome, but the DNA in it would not necessarily be exactly the same, because this could code for one protein that makes a certain eye color, and this could code for a different protein makes a different eye color, and it's the combination of the two that gives us our eye color. Same way with, if this is the gene that codes for insulin, how to make insulin, you'd have one here and one here, all right? And if one of those is off, that may be diabetes, all right? So, or it may be that both of them need to be off in order to create diabetes. So these chromosomes are called homologous chromosomes because they have the same genes on them, but they're not identical, all right? And those homologous chromosomes come from one from mom, one from dad, all right? So just to explain that, it's not going to become too important with mitosis, but it will with meiosis. These little things here are going to represent centrioles. Centrioles help a cell divide. They are only found in animal cells, so we are going to assume this is an animal cell as I go through this. So what I want to show you is what happens to these chromosomes during G1, S, G2, and through the stages of mitosis. And then you are going to draw them, all right, which I will show you here in a second. All right, so we're starting here in G1. So the cell was just formed, and when the cell is just formed, the DNA is in a form of chromatin, which just means it's like not really, you can't really see the individual chromosomes. It's just sort of all unwound and sort of a mess, all right? But notice again that my diploid number here is 6. I have 6 chromosomes in here, all right? It's just all wound up, all right? Inside the nucleus, here's the centriole sitting outside of the nucleus. All right, so getting ready then for S phase. In the S phase is when DNA copies itself. So only then do we get the other half of what we typically call the butterfly chromosomes. So during the S phase, this chromosome copies itself, all right? And so I have 2 identical pairs here of the same chromosome, all right? Same information. This is now called a duplicated chromosome, and that's the butterfly shape. All right, and each of them would do the same. So I'm going to duplicate each one. Duplicate, duplicate, duplicate. All right, so during the S phase, now I have duplicated chromosomes. But it's really still sort of all jumbled as chromatin, all right? And then during the G2 phase, it looks basically the same. Nothing really happens to the DNA, all right? So now we're getting ready, past all the checkpoints. Now we're getting ready to go into mitosis. So the first phase of mitosis is prophase. All right, prophase means like first, all right, which is why it's called the first phase. And basically what happens here is you will now start to see these duplicated chromosomes. They wind up into their chromosome form, all right? So remember, chromatin was just all unwound, and now they're wound up, all right, together, and you can see them as duplicated chromosomes for the first time. The other thing that will happen is the nuclear membrane will disappear, All right, so that way we can pull this DNA apart. So the nuclear membrane has to disappear to do that. And then the other thing that will happen is that these centrioles, one will move to the opposite side of the cell. All right, so we have just a couple things mainly that happen during prophase. We can see the chromosomes for the first time. The centrioles move to opposite sides, and the nuclear membrane disappears. That's the biggest things that happen in prophase. All right, now we move to metaphase. Metaphase to me is the easiest one to remember and the easiest one to recognize if you see it in a cell, because now these chromosomes are going to line up one on top of each other, all right, along the midline of the cell. Metaphase, midline, all right? They line up in the middle, all right? And what will happen with these centrioles is that there's microtubules, like fibers inside there, and they will start and they will come out. My strings aren't quite long enough to reach all of them, but they will come out and these strings will attach to the center of the duplicated chromosome. By the way, the center of the duplicated chromosome right here, like these white parts are representing, that's called the centromere. And that's just like it's another protein that actually holds the two sides together for right now. All right, so each of them has a centromere. All right, and that's where the microtubules from the centriole, I know a lot of central words, but that's going to go and attach to each of the centrioles. All right, so I can only do a couple of them because my string's not long enough, but the other side would do the same. All right, the other centrioles got microtubules, and they're going to come out and attach to the centrioles of the chromosomes. Okay? So imagine that this is attached here, all right, and attached here. I'm going to show you that here in a second. This sometimes is called the spindle, all right, where you have the centrioles and the attached microtubules that are attaching to each of the chromosomes. So think of like fingers coming out and attaching to each of the centromeres of the chromosomes. Notice that the way that I have the chromosomes lined up, it does not matter. You do not have to go blue, blue, red, red. All right, it can go in any order of those 46 chromosomes. Does not matter when you go through mitosis how they line up. All right, can line up any way. So that makes no difference because all we're going to want to do is split these copied parts away from each other. All right, during the next phase. All right, so they're getting ready here, all lined up in the middle, chromosomes attached. This are the centrioles attached, sorry, and that's called the spindle. All right, so this whole thing here with the microtubules and the centrioles attaching, that's called the spindle. Sometimes called spindle fibers too. All right, so now what happens is in anaphase, the cell is going to elongate. So imagine that if this string is tied onto this centromere here, and I take it and I pull it like this, it is going to pull apart these copies, and this side's going to do the same, and it's going to pull apart the copies of the duplicated chromosome, all right? And it's going to do that all along, all right, the line, because they would have been attached to all of them, all right? And so I'm pulling them apart, all right? So now I'm back into the single chromosome instead of a duplicated chromosome. I've pulled the copy away from the original, I guess, all right? And that's what we've done. So it doesn't mean that all copies will go to one side and all originals to the other, all right? Again, it's random. I don't know. It doesn't really matter because they're all just copies of one another. But anaphase is pulling apart of the duplicated chromosomes into single chromosomes, all right? And these spindles and the centriole is the thing that does that, all right, for us. All right, so they move away a little bit, all right? And so then anaphase is done. All right, and then the next thing, sorry, I dropped my centriole. The next thing is telophase. So telophase basically is that this then all gets into a nucleus, and this will form a new nucleus, and then new nuclear membranes will reform around this DNA. All right, so what we would have during telophase is a cell that actually has two nuclei in it with exactly the same information. So if you look at this, remember the copies separated. So here's the original, here's the copy, all right, have exactly the same DNA on both sides. Here's the original, here's the copy, exactly the same DNA on both sides, and so on and so forth, all right? So when we do this, when telophase occurs, what you end up with is two nuclei. We've essentially now split the DNA, the copied DNA, into two nuclei, all right, and to get ready for the cell to split. Exact copies of one another. All right, and then cytokinesis occurs. Remember, cytokinesis technically is not part of mitosis. And cytokinesis, now the cell is going to split. Now, that will happen two different ways with an animal and a plant cell. In an animal cell, remember its outside membrane is a cell membrane, which is pretty fluid and flexible. And that can happen by if this is the outside of the cell membrane, it can pinch in on itself like this and form two new cells. All right, so cytokinesis in an animal cell involves the cell membrane pinching in on itself and then splitting into two. With a plant cell, that can't happen because a plant cell has a rigid cell wall. So instead of pinching, all right, if this were a plant cell, it just has to form a new cell wall in between. All right, and that new cell wall is called a cell plate. All right, it's just basically a baby cell wall, all right, that forms, all right, until it matures and becomes a full cell wall, all right, and then you've split the cell into two. So that cytokinesis looks a little different in animal cells and plant cells, just because animal cells, the membrane is a little more flexible than the cell wall, okay? I was thinking of something else that I wanted to tell you. The other difference is that plant cells actually don't have centrioles. So they have microtubules like this that are in the cell, but they don't come out of a centriole. So centrioles are only found in an animal cell. So it looks a little different. The process is basically the same, just that end part and the spindle looks a little different because it doesn't come out of a centriole in a plant cell, and the cytokinesis occurs a little differently in a plant cell than an animal cell, okay? All right, so I'm going to go back up front, all right, and I would like you then to draw the stages, dropping my microphone, sorry, draw the stages of mitosis, all right? So let me flip my camera so that I can show you what I would like that to look like. Maybe I can flip my camera. There we go. All right, so let's cut out of this so that you don't see that. All right, so that's just the phases. All right, so what I would like you to do is to draw, let me just get rid of this. I would like you to draw mitosis. Let's do all of it. Let's do the whole cell cycle. All right, draw chromosomes. All right, and I want you to start with the diploid number of 6, just like I just did when I just showed you that with the little pop beads over there. Diploid number is 6. And what I would like you to do is draw them with 3 pairs with 3 different colors, just like I had the chromosomes there that I just showed you. All right, so basically you're going to draw a pair of, here I have, gray ones. All right, then you would draw a pair of perhaps yellow ones and a pair of purple ones. All right, so there's your 6. All right, so what you're going to do is show me what's happening to these chromosomes during G1. What does that cell look like? So you're going to draw a cell. You're going to draw a nucleus because it has a nuclear membrane at that point. And I want you to label these things. Here's my cell membrane, here's my nucleus. And you're going to show these inside this cell, right? So I'm sort of giving you what G1 should look like. All right, and gray. And they don't, I'm showing them beside each other, but I'll show this one over here. They don't have to be beside each other. They can be anywhere. All right, and next then you're going to show me S, right? What do these look like in the S phase? This is when they copy themselves. What will they look like during G2? What will it look like during prophase, metaphase, anaphase, telophase, and finally cytokinesis. So I want you to actually draw cells like this, labeling, like these are my chromosomes, this is my nuclear membrane, this is my cell membrane. All right, then do the same thing in the S phase. Now what does it look like? During G2, what does it look like? In prophase, remember I said the nuclear membrane disappears, so you can like show this like a dotted line, all right, to show that it's disappearing, or show it disappeared totally. And what would the chromosomes look like? What are the centrioles doing? So I could even draw centrioles here, all right? They sort of look like stars, all right, during G1, okay? What's happening to those? Where do the spindle fibers form? How they line up? All right, show them splitting apart, show them 2 new nuclei, and then show what would happen at their final split. What do the 2 new cells look like after cytokinesis? So you can do this on Notability if you want and turn it in. You can use the chalk markers to do it. You can even use the Play-Doh, all right, and make yourselves little chromosomes and show them moving, all right? If you want to do a video or just take pictures of each phase, all right, using Play-Doh. It's totally up to you how you want to do this. And you may work together to do it, that's fine, if you're doing something a little more than just drawing. But that's what I would like you to work on here, is to diagram the stages of mitosis. All right, so that's your job here for the remainder of the period, okay? All right, hope that helps. And I do have another video on Schoology that shows sort of an animated version of what the stages of mitosis look like, if that helps. That should be what you're going to work on today, okay? All right, I will see you tomorrow.